everyone and welcome to my YouTube channel Perfect Lab. Have you ever wondered how microbiologists grow microorganisms in the laboratory? It all begins with something called an inoculum. In today's video, we will learn what an inoculum is, why it is important in microbiology, and how it is used to start microbial growth and culture media. And I believe by the end of this lesson, you will understand the role of an inoculum and the basic steps involved in inoculating culture media using an inoculating loop. So let's get started. So before we look at what an inoculum is, I just want us to study this picture over here. The first picture over here is our source of microorganisms. In this case, we have bacteria culture, but like we can also have a fungi culture or a yeast culture, but just that for this pictorial explanation we are using a bacteria culture. And the second picture over here shows a small amount of the microorganisms or the bacteria culture over here picked with a sterile inoculation loop to inoculate a new sterile culture medium over here to cause microbial growth. So we define an inoculum as a small amount of microorganisms such as bacteria or fungi or yeast that is introduced into a new culture medium to start microbial growth. Let's look at the steps involved in inoculating a culture medium with an inoculum using an inoculating loop. The first step is to flame to sterilize the inoculating loop, and in doing this we must make sure that we flame the entire wire including the loop in the blue core of the benzene burner until the whole wire glow orange red. And once we are done with it, we allow it to cool for 5 to 10 seconds. This is because using it while it's still hot can kill the microorganisms that we want to inoculate. The next step is to gently touch the cold loop into the microbial source to pick a small amount of the inoculum. And once we are done with it, we inoculate the media using a proper streaking technique. And in doing this, we must make sure that we work near the flame or we work in an biosafety cabinet to prevent contamination. And once we are done with this, we flame the inoculating loop again to prevent contamination and to keep the loop sterile. Let's look at the simple steps for transferring an inoculum using a micropipet. The first step is to prepare the micropipet by setting the micropipet to the desired volume and attaching the sterile pipette tip to it. So once we are done with this, the next step is to aspirate the inoculum by pressing the plunger to the fetch top and gently immersing the tip into the microbial culture and slowly releasing the plunger to draw the desired volume of inoculum. And once we are done with this, we dispense this inoculum onto a new sterile media by pressing the plunger smoothly to the first stop to dispense the inoculum onto this new sterile medium. So if the medium is a plate, we spread the inoculum using a sterile spreader if needed. And if it is a broth medium, we dispense directly into the liquid. Once we are done with it, we eject the tip safely and then label the medium and incubate. So some key points to remember when transferring an inoculum with a micropipette is that 1. Always use a sterile pipette tips and always use sterile techniques. And also change tips between different samples and never touch the tip to any surface including the media. and always use the correct volume for accurate results. This is because using a micropipette allows accurate transfer of a small measured amount of inoculum whilst minimizing contamination and ensuring reliable and reproducible results. So thanks for watching, and if you found this video helpful, please like, share, and subscribe to Perfect Lab. Leave your comments and questions below, and don't forget to scan the code on your screen to follow us on WhatsApp for more laboratory updates and tutorials. See you in the next video.